Let's start the final part of the morning session. We have just 2 other ongoing talks before having lunchtime breaks. So let me introduce the next speaker. The next speaker is Chun Xu. He is from Institute of Neuroscience, Zilles Chinese Academy of Sciences, Shanghai, China. The title of his presentation is Target Pattern Dependent Emotional Processing in Hippocampal Circuit. So please welcome Chun.
Thank you. Thanks. First, I would like to thank Karl and Nicola for organizing this event. So I'm Chun Xu from Institute of Neuroscience in Shanghai. So today I'm going to talk about our the work, which is the title here is about emotion processing, but it's rather more specific questions. So I would like to start from some more general introduction from the hippocampus, which of course you already heard a lot. Now, just to get everybody on the same page, the hippocampus is what's page? This one. Big one. Okay. That work? Oh, okay, great. Yes. So the hippocampus is apparently very famous because of its patient HM. So we know that that's kind of a very critical structure for the learning and memory. And then we also have another work by Chun Xu showing the hippocampal neurons can encode the place very well. And you have seen these slides. And there's this structure is highly evolutionarily conserved, but still there's many variabilities. We know that. Great. But now for the rodents, we see this is a dorsal-ventral, this kind of a gradient is corresponding to this anterior-posterior axis in the primates. So before it's known in the hippocampus, there's not of a synaptic plasticity such as a long-term potentiation, which is hypothesized to be the neural substrate for the learning and memory. So if you look at the MAD receptor in the hippocampus, you will see the deficit in the spatial learning, for example, the automates. So that's all the classical work. Now, in terms of the heterogeneity in the hippocampus, if we broadly divide, this is about rodent, a similar conclusion, I guess. That's also drawn from the human hippocampus. So this is dorsal hippocampus is corresponding to posterior human hippocampus, and they have a distinct connectivity in the brain and also with the functions. So this is the so-called, it's kind of called the cold hippocampus. This is called hot hippocampus, which is has more relation with emotion. Well, we talk a lot about their difference, but they still have many something in common. For example, for the contextual field conditioning, so the both dorsal and ventral hippocampus have been shown. They are critical for the context processing, such as the contextual field learning. And if you look at the place cells, so this is work from like in the Moses lab. So they have found that the place cells have a certain variability along this axis of longitudinal axis, we can call that. They also have a variability along this proximal distal axis. So that's defined by the proximity to the dental gyrus. So you see the place information, spatial information of place cells in the C3 neurons also varies depending on their somal location along this pyramidal cell layer. Now, in terms of gene expression, we also see many variability and or diversity, if you say that. So we can further subdivide subregion of hippocampal subfields into several parts. So I don't have time to talk about this more in detail, but we do have an ongoing work to try to use a spatial transcriptome to differentiate the potential molecular basis for these spatial subregions. So we have a lot of understanding about hippocampus. For example, we know that's important for different type of functions. They have brain-wide projections. So if you wish, we could separate them into the intro and extral hippocampal formations. This is including the internal cortex and the other hippocampal regions. And we know these key circuits, which is the most famous one is the trisynaptic circuits, but they can also be more broadly connected with others, particularly the sububiqual regions. So, however, there's still many unknowns so far. For example, whether there will be any subdomain or subtypes of the hippocampal neurons, and particularly regarding their distinct connectivity and the gene expressions. I think the most unknown so far is at the single cell level, how do the individual neurons, they project to multiple targets? For example, the intra and extra targets, and also the brain-wide targets, and also what is the relationship between their somal location and their axon targeting patterns. And also, we know that hippocampal neuron have these cross-hemisphere projections. So what would that be, the organizing principles? So to address all of these questions, we need to first to have an understanding of based on the single cell resolution, what their brain-wide projections. And of course, this is going to bring a big challenge for us because we're going to handle a big data with this high resolution imaging, which is really at the micron resolution for these rodent studies, for example. And of course, we need to create a database and to do the data mining analysis. To achieve that, we will need to achieve a sparse labeling of these neurons. For example, here showing an example. You have this soma, you can sparsely label, and you can track their axon projections to very far away in terms of these downstream targets in the axon terminals. So we teamed up with a group of people, actually a group of labs, and we use this sparse labeling technique combined with so-called fMOS imaging. So this gives us very good 3D reconstruction of these pictures. And then we use this big data analysis, and then we can finally accumulate more than 10,000 hippocampal neurons. So this is for the mouse hippocampus. So all these neurons, you see the location of the somas cover all the subregions and also along all other axes, including the longitudinal axis. So you see this whole brain reconstruction of all type of neurons in different subregions. And this is one example neurons you can see. The soma, this is represented by this dark point, and you see the axons quickly diverged into two pathways, what we call the caudal and the rostral. The rostral pathways goes through the phonics, reaching to the prefrontal cortex and hypothalamus, for example. And these caudal pathways, they will go to the cortical regions and also the amygdala regions, for example. So to take a long story short, we divide these neurons based on the targeting patterns. So these still are relatively large brain areas. For example, we divide the isocortex depend on their projection pathway, caudal, rostral. And so we use these patterns, we can define more than 300 targeted patterns. Still, we use these other axon trajectories. So these are we use these human identified trajectories as a templates to make a digitalized reconstruction of each neuron's patterns. So in that sense, we can better quantify the similarity of the morphology of all these neurons in the certain target patterns. So further, then we can group them into about like 43 subtypes. So you see these different type of the axon trajectories this is the organization of these. They have different compositions. Now, putting all these neurons together in a big map, we can roughly divide these 10,000 neurons into three big categories. So, for example, the group 1, which is exemplified by these neurons, they show very typical projection to the hypothalamus and the striatum areas. So the group 2, they have a further additional projection to the olfaction areas. So the group 3, they have many actually as a bilateral projection. For example, they have projection to these hippocampal, which is like C3 neurons. And also some neurons send very typical projection to the isocortex and hypothalamus. So apparently there are many more details of these neurons, but then I would pick up some kind of examples to show you how this data we may get into, and maybe also for in terms of the functions studies. So first one, this is kind of a confirmation of these projection studies. So you see the soma in the hippocampus, the color-coded by this, and you can also see their terminals. This is terminal points going into the medial internal cortex and the lateral internal cortex. So this is really nicely topographic correlation. You see this PCA analysis showing this highly correlated, highly organized. So this is known based on the classical studies. Now, we can also put that in a more conceptual level. So you see at many different type of brain areas, and we split the hippocampal cells into 6 bins. You can see their gradual changes. So depending on their preference, for example, the neurons which is projecting to the so-called cortical subplate. This is many, including brain areas, including the amygdala. You see the projection is heavily originated from the ventral hippocampus, ventral C1 neurons. But then in the isocortex, you see many more neurons in the dorsal hippocampus. They send direct projections to the isocortex. So now, this is the same, the slide I showed before. Now, we have done a little bit analysis based on the spatial transcriptome. So since we have quantify these projection subtypes. So we wonder whether they show any correlation with these gene expressions. So these are a theory of sections. We have this stereo-seq technique. So with this in situ sequencing, we can know about like 500 genes at this moment. So they have a second generation, which is give us more genes. But with this already, we can separate them, do the unsupervised clustering you see here, and you can quantify these potential spatial genes, spatial transcriptome clusters showing different abundance. For example, we can roughly, you have especially with the cluster 1 and the cluster 2, you see this is kind of some abrupt changes indicating there will be some kind of a genetic basis for this so-called as a dorsal and the ventral hippocampus. Presumably, this is ventral and this is dorsal. So this is sorted by the Bregmas. Of course, the question is whether they show any correlations with our projection subtypes. So this is show their distribution along this Bregma anterior-posterior axis. You see some projection subtypes correlate very well with certain gene expression clusters. So this is one example, the density of the soma, and you see that this is a gene expressions. So then that's a single example genes, for example, in this case. So we can run through a systematic correlation analysis. We can find quite some projection subtypes correlated with certain gene clusters. Now, of course, we also see the heterogeneity of the expression of these receptors and ion channels, for example. So I think the message here is we do observe there has distinct gene expression profile corresponding to these presumably projection subtypes. But so far, we don't have the single cell resolution yet. So the future step would be we could achieve the single cell projection together with a single cell gene expression profile. So that will be hopefully really address this question with how the subdomains are reached with these projection subtypes. Now, what about the anatomic function heterogeneities? So we observe one thing which is very interesting is that we found many neurons actually have these bihemispheric projections. So here you see that for certain projection subtypes, many of them are showing these bilateral projections. Now, this is not surprising for the hippocampal CUC neurons. So this is kind of expected. Many of these bilateral projection neurons actually coming from the C3 regions. But we also see some neurons actually in the C1 region. You see this is a single example cell here. This is a soma, and they have a really bilateral projection with a kind of symmetric. And these cells are more enriched in the dorsal hippocampus. So that's, we still don't know the reason, but maybe because they're more closer to each other's. So this is another type of example. We see the neurons projecting to the, basically this is a projecting to the MEC. They are highly specific enriched to the layer in the MEC. We believe these neurons are coming from the subicular. So we hypothesize these neurons probably they are carrying the head direction cells, which could be very important to the generation of the grid cells in the MEC. Whether that's true or not, we have to find out by the functional recordings, physiological recordings. So we see more other type of bilateral projections. Now, the principle is that the majority of these neurons, they only choose one pair of downstream targets. So, for example, if you see neurons projecting to the nuclear combines, so they only have this pair of targeting. They would not have a simultaneous targeting to others, like the 18, for example. The only exception is the C3 neurons. So they always have a bilateral projection to the lateral septum in this case. So what's the function significance of these neurons? There was one study from our collaborator at Xunin's lab. So what they have done is to silence these neurons using optogenetics techniques. So they silence their axon terminals in the other side of the neurons. So they show that they will bring an impairment to the rapid context generation. So normally, when they have to context exposure first, and this will have a very quick context generalization, which is a sort of hypothesis and memory linking. But with this type of inhibition, they can impair that kind of rapid generalizations. So I talk about these intro and extro hippocampal projections. So we can basically divide the single cell preference based on their preference for the CA or in the LEC or MEC and so on. So how about their extra hippocampal formation projections? So here we also see some interesting preference or kind of a coordination. You see the neurons projecting the central amygdala, the CA fields are different from those ones projecting to LEC and MEC. If we calculate their collateral index, for example, with the single neurons, you can consider how many brain targets they innervate, and then we can calculate simply a collateral index. So you will see that the neurons preferentially targeting MEC, they tend to have a much lower collateral index compared to those neurons preferentially targeting LEC. So that suggests the neurons have a strong projection to LEC, the hippocampal cells. They tend to have a global broader casting of information to the brain wild targets. Well, those ones targeted to MEC, they are more focused, they are more dedicated to the MEC projections. Now, we can also have some correlation analysis based on the single cells. So here, you have a targeting to two brain areas simultaneously, but you can have a very different correlation. For example, you have the cells either both weakly or strongly innervated to targets at the same time. But you also have other situation. So they only strongly innervate one of them, so it can be opposite. So this is the color code showing that we have different targets innervating both the intro and extra, but with a very different correlation. So I think this tells us another point is we should not only look at whether a neuron project to two brain areas, but also look at their strengths, because that probably has more relation with their functions. So that's this is a kind of some new findings from this projectile analysis. Now, from those analysis, we also see some interesting targeted patterns. So this has to do with what included in my title. So here you see the single neuron examples. So for example, you have individual neurons preferentially targeting to either prefrontal cortex, basal amygdala, or nuclear accumbens. So we chose these three brain areas because they are these regions highly involved in the emotion processing. So you see neurons also have this preferential targeting to two of them, or even three of them. So their soma location also shows some preference. For example, the neurons which are only targeting the prefrontal cortex, in this case, they would rather close to the dorsal side, or you can call that intermediate axis of this longitudinal axis, which is represented by the dashed line. So for the neurons preferentially projecting the basal amygdala, they really down to the ventral side. Now, of course, you have this kind of mixture of these neurons. So this is something we are very interested in. What could be their functions in terms of these specific connection patterns? So to approach this question, we need to find a way to label these neurons. So the traditional methods, if you do a retrograde tracer, either with dye or with AAV, which is retrograde, you cannot differentiate this type of projections. Now, there is an intersectional strategy. For example, this is a tool developed by the Karl lab. So they can mix with the Cre and flippase or other molecular controller, and then you can have effectors. Depending on the, for example, Cre on, flippase on, then you may express a certain marker or options to do that. Now, the problem for these kind of tools in many cases, the expression is too low for optogenetics or calcium imaging, because in general, this type of experiments need high expression of those effector genes. So we developed new molecular tools for this. We split the TTA into two parts with a protein slicing, so with the help of this intron sequence. So if the intron sequence can be split into two N and C terminals, and then once we express in the same cell, they can bring these two parts together. So now the TTA can be functionally and then drive the effectors. So the effectors can always be the same because they always controlled by the TTA. Now we verify this strategy. So here, let's say we express the TTA N parts and the C fragments. So only together with both, that you can have the successful expression. For example, here in this case, a Chun Xu, and then you can drive the neuron to phi. So using this approach, so here shows the calcium imaging data. We targeted the neurons. So, for example, the neurons simultaneously targeting to the prefrontal cortex and the amygdala, or the neurons targeting to amygdala and the nuclear accumbens. So here you see different type of combinations. So they show strong calcium responses to either the strong full shocks, which is aversive, or to the circus, which is rewarding. So you see many neurons actually show responses to two of them, which is we consider as a cylinder detector. Now we also see some neurons, which is projected to the amygdala and the prefrontal cortex. So they show those small responses to the, for example, to one of the shocks, but they do not show much to this one. So, and then we also see these ones, nuclear accumbens and prefrontal cortex neurons projectins. They also see this as some kind of a different preference for these projectors. Now, putting all those kind of data together, we hypothesized that because of this calcium imaging data, the nuclear accumbens and amygdala co-projector, they tend to be function as a cylinder detector. Now, the nuclear accumbens and prefrontal cortex projector, they pretty much have strong responses to the negative stimuli. So we use optogenetics using the same strategy. So this is real-time place preference. So when animal can freely explore these two contexts, and you only stimulate one side. So if this is truly rewarding, they may tend to like to stay inside. Or if it's a negative emotion, they would try to avoid this. So we see that if we stimulate or bidirectionally or inhibit these cells, so the animals show this kind of different preference of the scores. So suggesting these neurons, they do have this bring negative emotions and induce this real-time place preference. So for this type of analysis, we can, of course, do more complicated patterns. For example, here we have a neuron, a single projection neurons. They project into 4 targets simultaneously. So we can use these tools in combination with other Cree and flip-based controlled. We can intersectionally, let's say here, we have 5 features. So with these neurons, we can see that they also respond to foot shocks. So that is something we're trying to propose to when we study this circuit function of these neurons. Before, we typically have a single target. So you do this retrograde tracing, and you end up with a certain group of neurons. Maybe they are separated already, and then we interpret them as a different functionalities. Now, in this reality, we very often now have this situation, a single neuron actually targeting multiple targets. So, for example, here we have the hippocampus and amygdala co-projecting neurons and other types. So I think this is going to be bring the next challenge for us for the circuit studies. Even more complicated, we need to differentiate these patterns not only by the projecting, but also by the brain area which they do not project to. So this is a more sophisticated patterns. You end up with some other on and off these strategies. So hopefully this can be at least partially by this kind of tools, intersection tools, we would understand more and more about the function diversity of hippocampal neurons. So in the hippocampal field, there is a very classical hypothesis or theories proposed by Per Anderson. So this is called the laminar hypothesis. So this is based on his electrophysiology recordings. When you stimulate the C3, for example, and record different sites in the C1, you see this very sharp tuning. Based on this, he proposed that the hippocampus is function as like a series of independent theories. This can be like the central processing unit, this is the CPU, or analog to the functional column in the cortex. Now, from these single neural projectons, we start to see a simple, for example, example neurons like this. You see these neurons, this is soma, the blue is the C3, and you see the red are the C1 axon arbors. Actually, this spans a lot, quite much wider than the so-called lamina. So that seems to be contradictory to this very classical hypothesis. So we decide to look at this, quantify their preference by this index. So we calculate this longitudinal component versus this transverse component. So if you see the component for the longitudinal, means this neuron go along this longitudinal axis, and then if this neuron the axon prefers the transverse direction, that's more consistent with the Per Anderson's hypothesis. Now here, this shows the color. So the red color, it means this transverse, the green color means the transverse orientation. The red color means the longitudinal correlation. So what you immediately see is this aligned to the soma. So that means right at the soma, their axons will go transversely. Now with the red color, actually, we see that they will go longitudinally. At the end of this traveling, you will see the green color pop up again. That means they will go then transverse again. And these C3 neurons, their axons, will go into the C1, which is, again, you see these follow this transverse axis. You don't see this is the red and then the green at the end. So to simplify that, I think the cartoon is like this. So the individual C3 neurons, they have this transverse orientation of the axons, end with the C1, but they also have this spreading collaterals along this longitude axis. And then they will go very sharply using the transverse orientation into the C1. So I think that's kind of explained consistent with Per Andersen's observation because they do the field stimulation. When they do the electrodes, you always have this transverse innervation. So that kind of fits well with the electrophysiological recording. But you don't know whether this axon is coming from the C3 neurons from the same lamina or from different lamina. So I think this really clearly tells us the hippocampal neurons, they organize in a very, very elegant way. They follow this lamina hypothesis, but also they have these longitudinal collaterals, which we have some ongoing studies to study these neurons in terms of their information processing about the context in the emotions and so on. So I think I'm running out of time, so probably I would skip this part of this function studies, which is very independent part, and then I would jump to my conclusions. So we have generated a database, and this is freely available to visualize, and everyone can download that. We have a GUI. You can interact to see the neurons types, which one you want. So apparently, this is done by a team of people. The students in the lab, Chun Xu is a key leader for this. And we have this the whole consortium with leadership under by Mumin and Yan Gang. And so we have another bigger website to accommodate all types of data, including functional MRI data and other sequence data. So anybody interested, feel free to explore that. So with that, thank you for your attention.
All right, thank you very much. So we have time for a couple of questions. If you have questions, please go to the microphone. Yes, please.
Hi, as always, very impressive.
Thank you.
I have a kind of a philosophical question. It's something that I've been grappling with for many years in the context of how many areas exist in the cortex. But now I see the field developing, and a similar question is appearing: how many different types of neurons exist in the brain? And I think it's a real challenge because, for example, in this case, I'll make a guess, you correct me if I'm wrong. You looked at all your mice were from a specific age?
Yeah, from adult animals.
Yeah, and they were in a controlled environment. So I think if you were, for example, for a wild population, there will be less or more specificity in terms of gene expression according to what the animal, some genes are like that. So do you have any thoughts about, is the field is moving in this direction? How do we actually define what is a neuronal type?
Yes, that's a very fundamental question. Thanks for bringing that up. So I think, yes, so we need to have a consensus on the cell type definitions. So different people may have different criteria. But one thing I think is depends on I would say depends on function we're studying. So that's probably the part where we can more better to reach the consensus for that. Now, when we define the cell types, either based on the connectivity or based on the gene expression, so I think that provides a certain specificity. And exactly for this reason, we need the functional studies to carry on to really identify which one matters. Or there might be different type of projection patterns, but they all function the same. I think that's possible. I think so. The same question applies to the genetic markers. So in the end, I think what we provide here is a basis or an entry point for the function studies, and then we need to wait some time to accumulate the data to really identify the key subtypes. Maybe we can simplify that. So now we consider all the brain targets, but it may be in the end, there is several key brain regions is enough to define the most important cell types. So that's what I'm thinking, but it can be changed, depends on our progress.
Yeah, I'm thinking a message, not for you, but for me, for everybody involved in this.
Yes.
I keep thinking, okay, but what if the gene expression changes? At what point starts being a different cell type? How can you know if a certain cell type doesn't have a different pattern of gene expression or plasticity or whatever? So I think we're all going to have to be grappling with this question over the next 10 years or so until some...
Yeah, we will have to see the data. Yes.
In the same way that cortical areas, like people try to say, this is the map. But then depending on what you're studying, what functional measure you're getting, you might get different ways to parcellate.
Yeah, I agree. Thank you.
Any other questions? Maybe I have one quick question. So you are just talking about some correlational structure between some gene expression, maybe some axonal topography. I'm just wondering, do you have any just idea about how or underlying mechanisms are associated in between? Because the genes are maybe encoding some protein.
Yes.
So there are some related with some biological substitutes. So do you have any idea about how these genes are related for forming this topography? So.
Yes, I think there are two aspects to address it. So the first one is about the development or about the morphologies. So right now we are combining this kind of the connectivity and the gene sequencing by the barcode tracing. So we could have the chance to identify the neurons with a specific connected pattern, and then we know the gene expression. So we do the genetic comparison. So we can see some neuron, which are, some genes associated with, let's say, dendritic growth, axon growth. For example, we have neurons have ipsilateral projection. We have a neuron with a bilateral projection, which will give you very different lengths of the axons and dendrites. So we see some correlation with there. So I think that will has to do with their development of this circuit formation, for example. Now, the second aspect is about the functions. So here we pay more attention to about their channels, receptors expressions, and for that, we could also have some, let's say, positive relationship between their firing patterns, the neural excitability, and the channel expressions. So that may partly help to explain the diversity of these neurophysiological excitabilities. And along with this line, we see there have different behavior impacts in terms of this memory formation, context information processing. So I would tend to think that in two aspects, and either one, for example, for the development side, we probably need to go younger animals to see whether those genes are functional. But what we observed in the adult stage, that's probably something. It's a footprint. It's a footprint after this circuit formation already. And if we are lucky, we may find something. But we already missed something, which is did not keep this gene expression all the life. So yeah, that still has to be investigated.
Do we have time for another question? The same rules apply to everybody. I've got a question about the control of once you've got your gene and it's been translated into a protein, but then we've got all of this post-translational.
Yes. Yeah, all kinds of regulatory control
and everything. What do we know about how that is controlled?
Yeah, that's actually. So I think that's kind of putting in simple words is very complicated because these have the local translation of all these proteins in the dendrites, and even more debatable in locally in the axon terminals. And in addition, there will be the transport of the mRNA to the axons. So the short answer is I think it's not clear yet.
Good.
Yeah.
So it means we've still got like our grandchildren and their grandchildren.
Oh yeah.
Still have some work.
It's going to be for decades.
This is a difficult problem as far as I'm concerned because I'm interested in the receptors and
Yeah.
so some people think that using gene expression as a proxy for receptor expression levels, distribution patterns, but it doesn't quite work that way.
Yeah, that's another issue we should keep in mind. So this has to be validated by other approaches, like what you have been doing. This is immunostaining or other in situ proteins. That's definitely we should consider.
No, definitely, we're trying.
Thank you.
Okay, thank you very much again for Chun for wonderful talk.